In our paper, we describe a non-contact vision-based method for monitoring respiratory rate and heart rate. Participants simulated different sleeping positions to test performance under various conditions. The current gold standard for sleep monitoring is polysomnography. To record polysomnograms, patients must sleep overnight in a sleep laboratory while tethered to multiple wires, belts, and tubing. This process is inconvenient and uncomfortable. Our proposed solution is to capture physiological signals unobtrusively using computer vision. We recorded videos in 5 different positions. Each video was 5 minutes long, and there were a total of 17 participants. Recordings took place in a dark room illuminated with an infrared light. Ground truth heart rate and respiratory rate were captured via polysomnogram. An infrared sensitive camera was mounted above the participant such that the head and torso were in the view of the camera. Points in the image were detected and tracked over time using the KLT tracker. Point trajectories were produced for 30-second windows, and a new estimate was produced each second. For respiratory rate estimation, the point trajectories were bandpass filtered. We performed decomposition using principal component analysis and ranked the signals 2 ways, by the peak-to-power ratio or periodicity, and by explained variance. The respiratory rate estimate is the peak frequency of the power spectrum for the top-ranked signals. For smoothness, we compare the estimate produced by each ranking and choose the closest to the previous estimate, defaulting to the explained variance ranking if no previous estimate exists. For heart rate estimation, we apply a high-pass filter. We again perform a decomposition using independent component analysis. We rank the components by harmonic periodicity, a variant of periodicity that accounts for harmonics. We combine the spectra of the top-ranked components to produce an average spectra. The heart rate estimate is the frequency with the highest power in its peak and harmonics. In these graphs, sample results are provided of estimates, showing that our system closely tracks changes in the respiratory rate and heart rate over a 5-minute period. The average RMS error across all positions for respiratory rate estimation was 1.12 breaths per minute with a mean percentage error of 3.4%. The average RMS error for heart rate estimation was 5.87 beats per minute with a mean percentage error of 5%. The authors would like to acknowledge the support of the Toronto Rehabilitation Institute, the Natural Sciences and Engineering Research Council of Canada, and Bressotech.